Thank you. So I'm going to take you underwater, and I'm going to take you into a totally different scale, and which is the coral reef system, but also a scale in terms of modeling. So during the past 20 years, the percentage cover and abundance of coral reef ecosystems have declined worldwide. This decline has been more intense in the Western Atlantic, which is here, the green one, which corresponds to the greater Caribbean Sea. And when coral reefs decline, this is associated with a loss in the coral reef, in the reef three-dimensional structure, which is the slide on the right, and the biodiversity that is associated too. So the structure of coral reefs is changing in response to persistent disturbances. And this shift is often associated with a change from hard coral dominated communities to macroalgae dominated communities. And initially, like 20, 30 years ago, it was thought that these macroalgae dominated communities were just phases that could be reversed. But now we know that they are becoming stable states. So besides this type of shifting or transition, as you call it more in the vegetation world, there are other types of transitions, which is from hard coral dominated to just sponges or a mix of sponges and macroalgae. So I brought here some sponges that are becoming abundant in some regions. So that brown thing that you see is a sponge. It's an excavating and crossing sponge. And this is the barrel sponge. So the drivers of these changes includes top-down forces such as overfishing and bottom-up forces such as eutrophication and sedimentation that together with local and global factors such as hurricanes and climate change are affecting in more degree the hard corals or scleractinian corals. So I want to give you here a point at what are scleractinian or hard corals. So these are the corals that are able to secrete calcium carbonate, and that per se, they actually build the coral reef. So when you lose that, that specific type of corals, the reef system basically declines. You lost complete complexity. So when that happens, due to all these factors, there is a proliferation of other organisms, such as the sponges or the macroalgae. Now, within the sponge group is the phylum Porifera. They are animals, but they look like plants. We have some subgroups that is what we call the coral excavating sponges or boring sponges, because they can bore into the calcium carbonate skeleton of the corals. And in the past years, there have been an increase of some of these species due to higher levels of pollution, coral bleaching, coral mortality, hurricanes. And it is predicted that as all of these sponges increase, also their bioerosion rates on the calcium carbonate will increase in this also more lower acidic environment. Now, within the excavating sponges, I'm going to present you one of the model species that we have worked with, which is this Cleona deletrix. It's an orange sponge, excavating sponge. People think they are corals. Other people think they are plants, but they are animals. And this sponge is a very destructive sponge of the corals, and they can excavate inside of the coral skeletons 10 to 12 centimeters. So on the right, if you take a hammer and a chisel, that's what we did with that coral colony. If you chisel in, you can see that the sponge tissue is in the middle. That's the orange thing. Then around the yellow is the coral tissue. So these species had an increase in the coral reef system in Florida Keys and the northern coral reef tract. And between 1996 and 2001, basically increased their abundances, individual sizes, and cover. So we wanted to know at some point how much these coral reef colonies, the builders of the reef, are shifting into excavating sponge reefs or shifting into that state. And to what level climate change might be influencing this shifting. So why we're interested in that is because as these sponges can bore and excavate the calcium carbonate, they basically decrease and balance the system and decrease the topographic complexity and the fish microhabitats. So the objective was to develop a state transition model from healthy corals to excavating sponges. So here I bring you to a very tiny low level in the scale, which is the coral colony level. So these coral colonies could be from 20 centimeters up to 1 meters. They are bigger ones, but that's the scale at which I'm working, centimeters to meters. And the idea was to evaluate how much, these excavating sponges could take over the current deteriorated reefs and under different climate change scenarios. And on the side, we were thinking like, could this sponge be kind of an indicator of climate change at some level? So, I think I put them the other way. So to develop this state transition model, the first thing was to obtain information of how this specific competitive interaction occurs just ecologically. So we took some information, 7-year data from San Andres Island, which is located here. It's a Colombian island, but it's in front of Nicaragua. Then we took some data from Panama before and after 2010, and between 2010 and 2012, when we have a very drastic bleaching event that kills almost everything on the reef. And what I wanted to do that was to get the parameters of how much corals would survive versus how many of these sponges will survive. And then we actually had very few sites that have monitoring over more than 5 to 10 years. And the site that had that information was Fort Lauderdale, Florida, here. So we have, through the Coral Reef Monitoring Program, photo quadrants and transects of like 10 to 20 square meter transects at different depths with coral colonies. So from there, I did a subset of 100 corals initially. Then I took actually another subset from the same program, but in the Virgin Islands, to test the model. So I'm going to bring you back here into, okay, what is this, the system itself? Sorry, which is this one. I don't know what they were in other order. But what we did was to observe 43 different interactions. And we can see here, this is the coral in yellow. This is the sponge. And we observed this at 6 months, 13 months, and 7 years, the same exact coral colonies over time. So with this, I was able to evaluate, and you can see through the photos that at the end, we don't have any coral. The whole sponge took that coral colony, that space. So with this, I developed the ecological state and the conceptual model, in which we have a healthy coral that, due to a specific disturbance, can transition into a deteriorated coral. Once the coral deteriorates or bleach, it's not necessarily dead, but basically it's under stress. And at that point, the larvae of the sponge can attach on top of the coral. It competes with the coral for the space, and then it can take over that specific colony. Now, each of this deteriorated coral with a sponge and a sponge, at any time they can die. The coral can die, the sponge can die, and what they become is just macroalgae. Now, to come back from that macroalgae states to a healthy coral, you need coral recruitment. So I developed the model with 10 years data over the transects, and initially, I was working all of this in Excel. Finally, I got into the same model. So the first thing that we struggled with was the scale, of course, because the STSIM model only gives you us up to square kilometers. So we tried to keep it at the minimum and just think that our coral colony was just that side of the kilometers, but in our mind, of course, was not. So I already chatted with Leonardo, and he's going to add the meters into the STSIM, which is great. Now, vegetation types, we just go with the sponge and the states class, just what we have in our system. Now, to model the bleaching events, we actually decide to use an analogy, your fire. So the same way you model fire on the forest, we model coral bleaching and coral mortality on our system. And we use just empirical data from NOAA or our own empirical data from the field to do that. So what we obtain from these kind of models is kind of like, what is the proportion of these coral colonies that are dying and changing into the sponges, for example. And a very important point here is 2005, because 2005 was one of the major bleaching events in the whole Caribbean Sea and Florida. And you can see here in purple that was the peak moment in which the corals got the sponge on it. And then the periods after that, we just have the corals basically converting completely into sponges. So with this, We model moderate coral mortality every 10 years. That was what was expected at that time. And what we can observe is here in the first event usually is the more drastic for the corals. Once you have a 10-year event, then the healthy corals go down, but then the sponges start picking up over this 100-year timeline. And what we have here, all these peaks, the black and the orange one, is the competition between macroalgae and sponge. So we are actually modeling the competition itself for the specific space. Now we can translate that into percentage cover, and you have, in this case, we model a reef that is mostly macroalgae. So how much of that healthy coral in blue gets down and how much of the space is being taken by the sponge. And because they are bioeroders, we can convert that into bioerosion estimates of the coral reef system, which can give us that over time a good idea of how much the reef is getting flat. Now we set up other mixed model in which you can alternate moderate and massive events of bleaching. A massive event basically kills everything, all the corals. A moderate might bleach them but don't kill them. So when we do that, we can have times over these periods if they are corals alive, assuming this model assumes that there is coral recruitment and that there is some corals left. So the same situation happens and you can estimate the bioerosion at that period. So from this type of model, we learned that moderate coral mortality can temporarily shift coral reefs, healthy coral reefs, into excavating sponge reefs. Now, when you have massive coral mortality, this becomes more permanent. And then the situation is that now you don't have any healthy corals, but you have sponges that, depending how much they compete with macroalgae, they could be balancing. So sometimes the sponges win, sometimes the macroalgae wins. And what is important from here is that, yes, we are a very simple, very small scale, but at this scale is really important because you can have key species that transform completely the ecosystem and that can bioerode the system very quickly. So we learned that this sponge is able to take over that deteriorated coral reefs because in the moment that we have bleaching, that when the corals are under stress is when the sponge have the highest reproductive peak. So all the larvae is on the reef as the corals are going down. So I wanted to bring this to a different scale. This was just a small first level of interactions. And I asked, okay, can we model this for all different species interactions on the coral reef? So we start working on these 0.75 square meter quadrats. And that's where Christopher, my student, came into the task to manually map every single interaction over 15 years. And he's going to talk about this model after this talk. So what are the challenges that we have at this moment? First, we are doing everything manually, identification of species manually on these photographs. So we want to automate this classification of species and species interactions. The improvement right now is that we have a great collaboration with some optic engineers, underwater optic engineers. So they are developing at this point a true color LiDAR and fluorescent imager that we expect will help us to basically identify the species based on color and texture underwater. Now, the long-term monitoring is still a challenge because we don't have many sites on the coral reefs where we have long-term monitoring. And when you have images like GIS images or other kind of images, they don't penetrate through the water. So it can tell you here is the coral, here is the seagrass, but it doesn't give you anything more at the species level. Now, for the STSIM parameters and what we want to do, and I've been chatting these couple of days with Leonardo and Colin, is that maybe what we need is actually a coral reef package in which we could change and add other different values or descriptive information that is important for us. Age is not important for us. We cannot age anybody in the reef, but patchiness and fragmentation of the reef is super important for fish habitat or lobster habitats. And then the species interaction modes that I would like to add is that when and you're going to see that with Christopher model, when we have different groups or species, if you change or if there is a new interaction, we have to add it. So we have to type every single one. So it will be cool to have, like, if, for example, you have 5 species or functional groups, that you could combine them very quickly and add the interactions to that. So currently, Our idea is to continue working in the Caribbean. We have two sites, Florida and the Virgin Islands, where we have more than 20-year monitoring data, but it's this kind of very, small quadrants. We have other Barbados with less than 5 years monitoring, but we are trying to generate a new site for the next 3 years with the imager and try to get three-dimensional coral reefs with a much better quality images that allow us to model in a different level. And the whole question we want to answer here is coral bleaching and the mortality of corals at the regional level generating the same habitat? Like, do we have only macroalgae in the Caribbean? Do we have certain key species that are maybe a refuge for fish or for other organisms that are of interest? And with that, I just want to thank all the institutions that have supported. And I would like to thank also Apex and Colin and Leonardo for just being there and always supporting these ideas, and everybody here for these great 2 weeks. So thank you. 2 weeks, 2 days.